We're in the library today because they're busy in the dining room. So today I'm going to go through lipid digestion and also talk about vitamins and minerals. So lipid digestion and absorption and also lipid transport is quite a complicated subject. It's quite a bit more complicated than proteins or carbohydrate digestion and absorption. So hopefully we can try and make it a bit simpler. So first of all, the first step of lipid digestion is emulsification. So we'll talk about that. So the lipids that we eat are mostly triacylglycerol, and another name for triacylglycerol is triglyceride. So those two are the same thing. Just make sure that you realize that. And also cholesterol. So cholesterol is a small part of the lipids that you eat. It'll depend on what you're eating. But usually you don't want to eat too much cholesterol, and we'll look at why that is. So all these lipids are non-polar molecules by definition. So if they're in an aqueous solution, they're going to want to clump together and form globules. So the flat globules that enter the intestines, they have to be emulsified, which means they're broken down into smaller droplets in order to provide more surface area that's exposed to pancreatic lipase. And bile salts are released from the gallbladder to help break up the flat globules into micelles. And these bile salts are derived from cholesterol. So they have a non-polar side and a polar side. So you can see in that diagram below, you've got your fat globule, and then you have the bile salt coming in, which has the hydrophobic and the hydrophilic side, and they're going to surround a smaller fat droplet and so create a micelle. And then we have, once we've emulsified those fat droplets, which is providing a greater surface area, we can now allow pancreatic lipase, which is released by the pancreas, into that intestinal lumen, and it's going to bind to the micelles along with colipase. So colipase is the cofactor for lipase. So if you look at that diagram on the right, the colipase is acting like an anchor, and it's binding to the micelles, and then the lipase can bind to the colipase and chop up those triglyceride molecules. Okay, so it's important that we also have colipase, because if we didn't have colipase, then lipase wouldn't have anything to hold on to. Okay, and so it's going to hydrolyze the first and the third fatty acids from triacylglycerol, and so you're going to end up with 2 fatty acids and a monoacylglycerol. And you've got Xenical, which is a medication, and that's an irreversible inhibitor of pancreatic lipase. It'll bind to the active site and form a covalent bond, which will make it inactive. Okay, so just so you guys know, this is glycerol. Glycerol has 3 hydroxyl groups on it. So this is the glycerol. And then you'll have your fatty acid chain. So this is long chain of fatty acids. And that last one, so this is your fatty acid, okay? And then if you had a triacylglycerol, you've just got your glycerol molecule and it's so each one of these fatty acids is going to make an ester bond with the hydroxyl group on the glycerol, and I'm just going to draw a zigzag line to signify this long carbon chain. So this here is a triacylglycerol. So you can just abbreviate that as TG. Okay, so if I write TG, I mean triacylglycerol, okay, from now on. Or you could write it as TAG. Okay, so either works. But I'll probably just write down TG from now on. And then, so what pancreatic lipase does is it's going to come in and break the bond here and here. So then you're going to end up with a molecule that's just that's only got one acyl, so it's a monoacylglycerol, or you could say monoglyceride. This is monoglyceride. Okay. So the difference between monoglyceride and triglyceride is that triglyceride cannot pass through the intestinal cell membrane because it's too large. Okay, but monoglyceride is small enough to diffuse through the cell membrane of the intestinal cell or the enterocyte. Okay, and if you have a fatty acid, and I'm going to abbreviate fatty acids as FA from now on. Okay, so fatty acids can also pass through the intestinal cell membrane. Okay, so that's the whole reason why we need pancreatic lipase, in order to break down the triglycerides so that it's small enough to get through the intestinal cell membrane. Okay. So for the absorption of lipids, the fatty acids, monoacylglycerol, and cholesterol. So cholesterol can just diffuse across the apical membrane of the enterocyte without being broken down further. And there are also fatty acid transporters on the membrane. So if you remember the structure of the cell membrane, these are your phospholipid heads, right? And the phospholipids have their tails, and so it's cell membranes are phospholipid bilayers, right? So phospholipid bilayers. So there's two layers of phospholipids, and this side, remember the phospho heads are aqueous. They're polar, so they'll associate with the aqueous environment. So because this region is polar, the heads, and then the region in between, that's nonpolar. So the majority of the cell membrane is actually nonpolar, which means that the nonpolar molecules can simply diffuse across as long as they are not too large. Okay, so triglycerides are too large to simply diffuse across. Inside the enterocyte, the fatty acids and the monoglyceride get put back together into a triglyceride, and they're packaged with cholesterol into a lipoprotein called chylomicron. Okay, does everyone know how to spell chylomicron? So chylomicron. So a lipoprotein, I just like to think of it like a bus for lipids. Okay, so you need to remember that blood is an aqueous solution, so it's polar, and so it can't carry a lot of non-polar molecules like lipids unless they're packaged in lipoproteins. So lipoproteins are like buses for lipids, and lipids are just going to catch a ride and travel around in the blood inside the lipoproteins. Okay, and chylomicrons are one of those lipoproteins. These chylomicrons, once they're made in the enterocyte, they're going to be exocytosed into the lacteals present in the villi. So I'm guessing you guys would have learned about the lacteals in hubs, right? So they're the lymph capillaries, which collect the chylomicrons, and then they're going to collect in the cisterna chyli, travel up through the thoracic duct, and then empty into the junction of the left subclavian vein with the brachiocephalic vein, and then that's going to empty it into the superior vena cava, right? So when these chylomicrons, they're going to enter into the bloodstream, they're going to travel through the arterial system where the triglycerides in the chylomicrons are hydrolyzed by an enzyme called lipoprotein lipase. Okay, so that's lipoprotein lipase, or LPL for short. Okay, so it's a lipase that's acting on lipoproteins. Okay, so that one's an important one to remember, and that's present on the capillaries of your tissues, such as your adipose tissue. So adipose means fat, heart and skeletal muscle. So the lipoprotein lipase requires a cofactor, and that cofactor is called ApoC2. Okay. So the lipoprotein lipase hydrolyzes the triglycerides into glycerol and 3 fatty acids. So that's the key difference between pancreatic lipase and lipoprotein lipase. Remember that when pancreatic lipase broke up the triglycerides, what did we get? Yeah, you got a monoacylglycerol and 2 fatty acids. So lipoprotein lipase is going to cleave all 3 of the fatty acids off triacylglycerol. So you get a glycerol and 3 fatty acids, which are going to be absorbed by the tissues. Okay. So if the fatty acids and the glycerol is taken up by the fat cells, they're just going to resynthesize the triglycerides and store them as fat. And if they get taken up into muscle cells, the most likely endpoint for them is going to be they're going to be broken down by a process called beta oxidation. So we'll go through beta oxidation probably this week or next week for you guys. Not too sure, but you will learn more about that. and they'll be used for energy. So they'll be broken down into two carbon units, which are called acetate, and turn into acetyl-CoA, which will then enter the citric acid cycle. So once these chylomicrons are processed by lipoprotein lipase, they're now known as chylomicron remnants. Okay, so remnants just means the things that are remaining. So because most of their triglycerides have been taken out, they're now smaller and they have a lower triglyceride content. And remember that the chylomicrons initially had cholesterol in them as well, but the cholesterol doesn't get taken out of the chylomicrons. It's just the triglycerides. So now they're going to have, proportionally, they're going to have a larger cholesterol content, not because they've increased their cholesterol content, just because there's less triglycerides now. And these chylomicron remnants are taken up by the liver. Okay, and so there's LDL receptors and ApoE receptors, and they can bind to a protein which is present on the chylomicron. That protein is ApoE, and that binding has to occur in order to endocytose the chylomicron. So I'd just like to draw all of this out because I know it's quite confusing. So first of all, you have your enterocyte. So here's your enterocyte with the microvilli. I'm just going to draw one enterocyte. And inside so once you have this is the monoacylglycerol or the monoglyceride, which has come in, and you've also got your fatty acids. So they're going to be put together into triglyceride, right? This is happening inside the enterocyte. So this is your enterocyte. And this is going to be packaged inside a vesicle called a chylomicron, which is a type. So a chylomicron is a type of lipoprotein, and a lipoprotein is just something, a vessel that's going to carry around lipids. And this lipoprotein has several proteins, okay? And so remember how I said the cell membrane was a phospholipid bilayer? This is a phospholipid monolayer, okay? So these phospholipids, there's only one layer, right? So that means that, remember these tails are nonpolar, so that creates a nonpolar environment, for all these lipids inside, whereas the heads can interact with the aqueous environment. Okay, so inside your chylomicron, you're going to have a lot of triglycerides and a few cholesterol molecules as well. And so we're going to identify what some of these proteins are. So this one here, so we call, if they're part of a lipoprotein, these are apoproteins. Okay, so this is ApoE. Okay, so a chylomicron will have an ApoE. It'll also have an ApoB48. Okay. And it will have ApoC2 as well. So the function of ApoE is for binding So this will bind to ApoE or LDL receptors on the liver. And remember that once it becomes a chylomicron remnant, it's going to become endocytosed by the liver. So it's going to be cleared from the blood. And the function of ApoB48 is simply structural. So it just helps to maintain the integrity of that lipoprotein. Now, ApoC2 is probably your most important one to remember, because ApoC2 will act as a cofactor. So I mentioned this before. It's the cofactor for lipoprotein lipase. Okay, so this is for your chylomicron. So this is your chylomicron. Okay. So then it's going to, this chylomicron is going to enter the lacteal. It's going to enter the lacteal. And this is your, so now we're in the capillaries. So you might have your, here's your fat cells. So here's your fat cells, or you could call it your adipocyte. So adipocyte is the medical name for, well, the proper name for a fat cell. And you can also have your muscle cell, or if you want to be proper, you could call it your myocyte. Okay. So on the walls of the capillaries, you're going to have an enzyme, and this enzyme has a little groove. So this enzyme is called Lipoprotein Lipase. Okay. So you're going to have your chylomicron come into the bloodstream, and it's going to travel into the capillaries, and this apoprotein is going to bind to the lipoprotein lipase, right? So what is this one going to be? Yep. So it's going to be ApoC2. So ApoC2 is the cofactor for lipoprotein lipase, and it's going to bind to the lipoprotein lipase enzyme. So that's going to hold the chylomicron in place, so that the lipoprotein lipase can catalyze the reaction and break down those triglycerides. And it's going to break down those triglycerides into, so we said it's going to take all three fatty acids off that triacylglyceride, and you're also going to have, so that means you're going to have the triglyceride turning into glycerol and three fatty acids. Okay. And so the glycerols, these can diffuse into the fat cells, and so can the fatty acids. Okay, so these will just be turned back into triglycerides in the fat cells, and if they come into the myocyte, the fatty acids will be turned into acetyl-CoA, and this is through a process called beta oxidation. Okay, so that's so you can see that for this to occur, for the lipoprotein lipase to break down the triglycerides inside the chylomicrons, we need to have this ApoC2, and if you have a mutation in the gene for ApoC2, then you won't be able to bind to lipoprotein lipase, and that will mean that you're not able to clear these chylomicrons, and that's one of the gene defects that was mentioned in the lecture. Okay, so assuming that everything's all good, now it's been processed by lipoprotein lipase. Now what is it called? What does it become? So it's a chylomicron, and it becomes a chylomicron remnant. So it's a bit smaller now, and so this is a chylomicron remnant, and it's got, so what actually happens is that it's still got its ApoB48. So remember that the ApoB48 was a structural protein, so it just helps to maintain the shape. And this one here was ApoE. Okay, so ApoE's the one that's going to bind to the LDL receptor or the ApoE receptor on the liver, but now it's lost its ApoC2. Okay, so the chylomicron remnant no longer has the ApoC2, and so now it'll come to the liver, and there'll be ApoE receptor on the liver, so it's going to bind to that ApoE receptor on the liver. ApoE receptor, or it could be an LDL receptor as well, and then that's going to be taken up by the liver and just broken down, and there'll be a little bit of triglycerides left over, and you'll also have some cholesterol that was in that chylomicron. Okay, cool. So we'll move on to the next page. So chylomicrons are produced by enterocytes. Okay, so remember that chylomicrons are produced by enterocytes. They are not produced by the liver, and they're only present in the blood after eating a meal containing fats. Okay, so if you ate a meal and you only ate proteins and you only ate carbohydrates, you would not have any chylomicrons in the bloodstream. Okay, chylomicrons are 90% triglycerides, so they have the lowest density among lipoproteins. They are also the largest in size among the lipoproteins. Okay, so as a general rule of thumb, you can assume that lipids are low density and proteins are more dense. Okay, so if you have a chylomicron, it's mostly lipids, so it's got a little bit of protein, but compared to your other lipoproteins, it's got the highest fat content and the lowest protein content, which means that it's the lowest density, and it's also the largest in size. So one way you can see this is that if you have, so on the first page we talked about Xenical, right? So that's the inhibitor of pancreatic lipase. So if you inhibited pancreatic lipase, would you be able to absorb lipids? No. So you wouldn't be able to absorb lipids because if you have no pancreatic lipase, all your triglycerides can't be broken down into monoglycerides or fatty acids. So all of that triglycerides is just going to go through your intestines and come out in your poo, okay? And if that happens, there's a number of things that happen to your poo. And the first thing is that they look white because fat is white. Okay, so you're going to get these white poos. And also, they're going to float because they're full of fat. And like I said, fat is low density. So you'll get these white poos that are floaty. And no matter how much you flush, they're just not going to go down. Okay, so it can be quite inconvenient. And that's one of the reasons why Xenical, that inhibitor of pancreatic lipase, was not so popular, okay? And it also makes your poo stink, because if you've got lots of fat going into your large intestine, all those bacteria can just feast on that fat, okay? So you might have thought like, oh, this Xenical, I can eat as much fat as I want and I'll just poo it out. Sounds like a great idea, but it's actually not, okay? So I wouldn't recommend it. Not that I've tried it, I'm just, yeah, that's just how it is. Okay, so the last bullet point of chylomicrons, you've got, so you've also got ApoB48, which is only present on chylomicrons, and that's just a structural role. So, yeah, ApoB48 is called ApoB48 because it's only the first 48% of another apoprotein called ApoB100. And so ApoB100 normally binds to the Ldl receptor, but but ApoB48 can't bind to Ldl receptors. So then we have very low density lipoproteins, or VLDLs. So these are synthesized by the liver, and they can also contain a high proportion of triglycerides with a little bit of cholesterol as well. And they're a little bit denser than chylomicrons because they don't have as much triglycerides, but they're not as dense as low density or high density lipoproteins, which should be pretty obvious looking at the names. Okay, so VLDLs are very similar to chylomicrons, except that they have a slightly lower triglyceride content and are made in the liver. And their job is also to deliver triglycerides to the peripheral tissues. They also have ApoC2 and ApoE, but they have ApoB100 instead of B48. And these VLDLs are processed by the same enzyme that processed chylomicrons. So what was that? What enzyme processed the chylomicrons? So it was on the wall of the capillaries. Yep, so lipoprotein lipase will also process VLDLs. And once they get processed, they're going to turn into low density lipoproteins. Okay, so I think I'll just draw this all out again, but this time we'll look at the path of a VLDL. So if you go on to the next page, there's space for you to draw this out. Okay. So you have your liver. Okay. So this is your liver. So your liver normally contains some triglycerides. Okay, so you've got a lot of triglycerides in your liver, and you've also got some cholesterol, and you want to get rid of it. Okay, so the liver wants to get rid of its triglycerides and cholesterol and send it out into the bloodstream and send it out to the other tissues of your body. So what it's going to do is it's going to put all of it into a lipoprotein, and this lipoprotein is your VLDL, or your very low density lipoprotein. Okay. Okay, so it's also, so all of your lipoproteins are phospholipid monolayer, which means that the interior of this lipoprotein is going to be non-polar because it's carrying triglycerides. So mostly triglycerides and a little bit of cholesterol. And this is your so this is your VLDL. And so this apoprotein here will be ApoE. Okay. This one here is ApoB100. And this one here is the one that binds to lipoprotein lipase. What's the cofactor that binds to lipoprotein lipase? Yeah, so ApoC2. If you're going to remember one apoprotein, make sure that it's ApoC2. Okay, so that's the one that's going to bind to lipoprotein lipase, and that's present on both VLDLs and chylomicrons. So your liver's made this VLDL. Now it's going to go into the bloodstream. So it's going to go into the blood. Go into the blood, and on the walls of the capillaries, you have this protein here. So this is your lipoprotein lipase, and it's going to bind to this thing here, which is ApoC2, but this time it's a VLDL. So remember that lipoprotein lipase can bind to both VLDLs and chylomicrons because they both have the ApoC2 apoprotein. And again, you're just going to have the triglycerides being turned into glycerol and the 3 fatty acids. Okay, and again, these can just diffuse into your fat cells or your muscle cells, and they can be used for energy or just stored as fat. Okay, so once the VLDL has been processed, what does it become? So technically it becomes a VLDL remnant or IDL, or an intermediate density lipoprotein, but we're just going to simplify it a little bit, and we're just going to say that it turns straight into an LDL. Okay, so it turns into an LDL lipoprotein. Okay, and these LDL lipoproteins are very small. And nearly all of the triglyceride has been taken out of this LDL. So the main lipid that it contains is cholesterol. So it's mainly cholesterol. Okay. And it's lost the ApoC2, and all it has is the ApoB100. Okay. And so these are going to come back. So your blood. So here's your loop. So on your liver, you have your. This is your LDL receptor. Okay. So the ApoB100 will bind to the LDL receptor, and then that'll be taken up by endocytosis. Okay. And also, what can happen to your LDL molecules is that you'll have cells like ovaries and your testicles, which are cells that produce steroid hormones, and also your adrenal cortex can actually produce some steroid hormones as well. So if you remember that steroid hormones are lipid, right? So they're made from cholesterol. So these are the ingredients for your steroid hormones. So if you've got, let's say, a testicular cell or an ovary cell, they're going to want cholesterol in order to make their steroid hormones. So they will also have LDL receptors. So these LDL receptors can bind to the LDL molecules, the ApoB100, and then they can take up that cholesterol to. So this, you'll have this cholesterol, which is turned into steroid hormones. So that's things like estrogen or testosterone and other things like that. Okay. And then one last thing, so we talked about chylomicrons, we talked about VLDLs, we've talked about LDLs, and now we have HDLs, or high density lipoproteins. So high density lipoproteins are different. They actually have like a scavenging function. So HDLs are mostly proteins. So it's mostly proteins. So they're going to come out and they're not going to carry anything. But if you have a lot of cholesterol in your blood, what can happen over time is you can get a buildup. You can get a buildup of cholesterol on the walls of your arteries, and this is called. So this cholesterol plaque, and we call this atherosclerosis. Okay, and this is, if you look at any like 60 or 70-year-old's heart, especially if they've been smoking and drinking a lot and had diabetes or whatever, they're going to have like these big plaques on their arteries, and when these arteries when these plaques burst, that's when they have a heart attack, or these can come off. These can become dislodged and then travel into a small capillary in your brain and get stuck, and that's going to stop the blood flow to your brain, and that's how you have a stroke. Okay. So what HDL does is HDL you've got your HDL molecule. So it's going to pick up some of this cholesterol in the blood and take it back to your liver. Okay, so it's kind of cleaning up all of the cholesterol that's in your bloodstream. Okay, and that's why they say that you don't want a lot of LDL in your blood. If you have a lot of LDL in your blood, that's not very good, because some of this cholesterol can leak out and get deposited on the walls of the arteries and cause atherosclerosis. Whereas you do want to have a lot of HDL, which is the good cholesterol, because you're going to remove these cholesterol plaques. Okay, so if we go back to the page just before, we'll just finish it off. So low density lipoproteins contain mostly cholesterol. So low density lipoproteins contain mostly cholesterol. And the only apoprotein that they have is ApoB100. So remember, ApoB100 is the protein that's going to bind to the Ldl receptor. And because most of their triglyceride has been removed, their main component is now cholesterol esters. So you do need cholesterol for membranes and steroid hormones, and some of those. So LDL can be taken up by the cells that do need cholesterol, like testicles and ovaries, and LDL can also be cleared by the liver. And then you've got your high density lipoproteins, or your HDLs, which are small lipoproteins that are mostly protein. So they are the densest lipoprotein, and their function is to take excess cholesterol from cells and artery walls back to the liver, where they can be excreted into the bile. So remember that bile salts are also made from cholesterol, and they mostly ApoA apoprotein. So next page again. So familial hypercholesterolemia and other lipid disorders. So if you had a mutation in the gene for the LDL receptor protein, so this LDL receptor protein, if it was mutated, it's not going to bind to the ApoB100 on your LDL. So that means you can't clear the LDL out of your bloodstream. So that all these LDL molecules are just going to be stuck in your bloodstream, and they're just going to circulate, and eventually it's just going to turn into these cholesterol plaques. And you can also have a mutation in the gene for your ApoC2 protein, and or the ApoE protein. So that's going to cause a high level of triglycerides in your blood, right? Because if the ApoC2 can't bind to the LPL, then you're not going to remove the triglycerides from your chylomicrons or your VLDLs. So triglyceride can also cause these plaques to form. So LDL or triglycerides can cause atherosclerosis if there's too much in the bloodstream. And I explained to you guys what atherosclerosis was. Okay, so we'll move on to vitamins now. So we'll go through these a little bit more quickly. So vitamins are organic molecules. So organic just means they're carbon-based, that are needed in the diet because our body can't synthesize them. They don't provide the body with energy. So the fat-soluble ones are A, D, E, and K, and the water-soluble ones are B and C. Okay, and if you have a low level of vitamins, that's going to result in symptoms. Okay. And you can detect vitamin deficiencies by examining the person, doing blood tests, and measuring their body dimensions, and also looking at their diet. So the B group vitamins are important for energy metabolism. So most of these vitamins act as cofactors or coenzymes for all the reactions, all the enzymes involved in energy. So B1, who knows what B1 is? So B1 is thiamine. So thiamine makes up a component of thiamine pyrophosphate, which is a cofactor that's part of the pyruvate dehydrogenase complex. So when you convert pyruvate to acetyl-CoA, so you have this, you have an enzyme complex called pyruvate dehydrogenase complex, and that includes thiamine pyrophosphate, okay? And if you have a deficiency of thiamine, you're not going to be able to catalyze this reaction, so you're going to have an increase in the levels of pyruvate, and that means that you're going to have an increase in the levels of lactate or lactic acid. And if you have a lot of lactic acid, that's going to lead to some symptoms. Okay. B2 on the next page is riboflavin. So B2 is riboflavin, and that makes up FAD and also FMN as well. So FAD is critical for the citric acid cycle as an electron carrier. So FAD will pick up 2 protons and 2 electrons and turn into FADH2. Okay. And you'll learn more about this later on. B3 is niacin. So niacin is part of NAD+, or nicotinamide adenine dinucleotide. So this is one of the most important ones. You'll hear this over and over again. It's also an electron carrier. And this one's NAD+ picks up just 1 proton and 2 electrons and becomes NADH. Okay. So, just look at these molecules and make sure you remember what the functional groups are. So, which one is niacin? Okay. B5 is pantothenic acid. So it's written below in that diagram if you want to see how it's spelt. So it's either pantothenic acid or pantothenate. So that's making up coenzyme A, which is carrying acyl groups. So you can see that on the left-hand side, you've got that sulfur molecule. So that's the part. So the S on the left-hand side is the one that's going to connect to the acetyl group and the acyl groups. Okay. So usually you'd just say acetyl-CoA or acyl-CoA, but remember it's the sulfur on the end of the coenzyme A that's going to be the functional group, which is binding. B6 is pyridoxine. So pyridoxine, pyridoxine, and that's pyridoxal phosphate, which is another cofactor involved in transamination. B9 is folate, which is required for the proper synthesis of DNA and RNA. And then you've got B12. And if you ask me whether you need to know these, yes, you do. You need to remember what B1 is, what B2 is, what B3 is, because it does come up in the exams, okay? And you need to be able to recognize the structure of them as well. You don't have to be able to draw them, but you need to be able to see a picture and recognize them. So minerals are just non-organic elements. So quickly, what are some examples?
Magnesium.
Yep.
Calcium.
Yep. So sodium, potassium, and the ones that you just said. Okay. So they're not a source of energy, and you only need small amounts, milligrams or micrograms. So magnesium was mentioned especially in the lecture. So it's a cofactor for over 300 enzymes, and you can see all the stuff it's involved in. So now we'll just finish off with some questions from past exams. So like I said, you need to recognize, be able to recognize each structure. So you have three options there. You've got either NAD or FAD or coenzyme A. So what's the top one? Anyone want to have a guess?
Coenzyme A.
Yep. So the top one is coenzyme A, and you need to circle the functional group that's responsible for the carrier activity of that coenzyme. So which part would you circle? Yep, so you just circle the SH part. Remember that it's the sulfur that's going to bind to the acyl groups. So that's the structure, that's the group that's responsible for the carrier activity. And then the bottom left, what's that? NAD. Yep, so that's NAD. And which structure on that would be responsible? Yep, so it's going to be the niacin. So on the top, you have this, the N. So this whole thing here is your niacin, which is your functional group. Okay, so that's the part that's going to pick up the electrons. And so that means the one on the bottom right must be FAD. Okay, and that whole ring structure, the three rings, that's your riboflavin. Okay, and that's what's going to carry the electrons. Okay, so that one's pretty easy to recognize because that's pretty distinctive. So number 6, which statement regarding lipoproteins is incorrect? So A. So is A true or false?
True.
Yep, A is true. B. Yep. So if they're fat soluble, it'll be carried by lipoproteins. C.
True.
Yep, true. So VLDLs and chylomicrons are metabolized by lipoprotein lipase. And D, is it true or false?
True. False.
Yep. So what's false about it?
They.
Yeah. So lipoproteins are phospholipid monolayers. Okay, not bilayers. So if it said they consist of phospholipid monolayer surrounding a triacylglycerol core, that would be correct, but it's not a bilayer. 7, which one of the following symptoms would you not expect to see in a patient with ApoC2 deficiency? So if you had an ApoC2 deficiency, you'd have a lot of triglycerides in your blood, right? Because you can't remove the triglycerides from your chylomicrons or your VLDL. And if you have lots of triglycerides in your blood, you get pancreatitis. That's one of the complications. So you would expect to see that in a patient with ApoC2 deficiency. B, would you expect to see that or not? No. you'd have a high level of chylomicrons in the plasma because they're not getting removed. Okay, and you'd also have fatty deposits on the skin, so those are called xanthomas, and you'd have high levels of triacylglycerol. Next page, you have to list each of the coenzymes with the vitamin from which it is derived. So FMN is flavin mononucleotide. So which vitamin has flavin in it? Yep. So FMN would be number 2. NADPH, what would that be? Yep, so niacin, 4. And coenzyme A? Yep, so that's pantothenic acid. And TPP is thiamine pyrophosphate, so that should be pretty easy. Yep, number 5, thiamine. Okay, and you should also remember the number of those as well. So thiamine would be B1, niacin is B3, and so on. So if you have chylomicrons in your plasma after not eating for 10 hours, that would indicate a deficiency in A, pancreatic lipase. So if you had a pancreatic lipase deficiency, would you have chylomicrons in your blood? No. Because, why is that?
It's later down the track.
Yeah. So remember that if you don't have pancreatic lipase, none of your fat's going to get into your enterocyte. And if none of your fat's getting into your enterocyte, you're not going to make any chylomicrons, because remember that chylomicrons are made by the enterocyte from the fat that's come in. Okay, so if you have a pancreatic lipase deficiency, none of your triacylglycerides will be getting into the enterocytes in the first place. B, if you had a deficiency in ApoB100, that's the protein that binds to the LDL receptor, right? So that means that you'd have a high level of LDL in your blood, but it wouldn't affect your chylomicrons. Okay? And C is an unrelated protein. So D would be the correct answer here. So if you had a ApoC2 deficiency, you wouldn't be breaking down those chylomicrons. Okay? 7, which one of the following statements about vitamins and minerals is incorrect? So A, they're required in milligram microgram diets, amounts in the human diet. That's true. And is B true or false? Yep, so B is true. C. So they do have an important role as coenzymes and cofactors, but sometimes they can be structural as well. They're not always just coenzymes and cofactors. So for example, calcium plays an important structural role in your bones and teeth. And it's true that deficiency is primarily diagnosed by blood tests and symptoms. So thiamine, niacin, and riboflavin are all involved in energy metabolism. Okay, so A is true. B is not true because they are essential organic nutrients, and in the normal New Zealand diet, they're normally adequate. And D is not true because remember that the B and C vitamins are water soluble, not fat soluble. Okay, sorry I went over time a little bit today. It's 8:05. Hopefully you found that helpful. So thank you guys. We'll be back in the dining room next week. All right, cheers.